Welcome for the topic ovulation. Today we will see what are the component or factors, hormones that occur during the process of ovulation. So ovulation is a complex process. Ovulation simply means escape of oocyte from the dominant follicle. Suppose this is a dominant follicle and this oocyte is released from the follicle by digesting this apical portion of the follicle. We know that folliculogenesis occurs in the cortex or superficial part of the ovary. So digestion of this part is very important to escape the oocyte from the follicle. So it is a rupture of apical region of the follicle so that the oocyte is released. And successful ovulation not only folliculogenesis but also developmentally competent oocyte production also necessary. This is the oocyte which develop along the folliculogenesis. So therefore, along the folliculogenesis, the oocyte development or competency is also very necessary for successful fertilization or embryo production. So it is a complex process involving endocrine or coordination of endocrine with ovarian hormones or cells, autocrine function within the follicle. This we have granulosa cell and theca cell and oocyte. All these coordinations are very important for successful ovulation. So there within the follicle there will be inhibitory or stimulatory signaling pathway and hormonal that influence the resuming meiosis 1 especially, then release of competent oocyte for successful fertilization by degrading the apical layer of the follicle. Let's see how the process begins. So let's refresh ourselves. What are the structure in the follicle? So these are the theca cell and the basement membrane, then granulosa cell, then fluid-filled cavity, we call it antrum, then the membrane or layer around the oocyte is called zona pellucida, then red color is the oocyte. So we have theca externa, outside is externa, then the theca cell which are close to the basement membrane, these are called theca interna. We know that granulosa cell, they have the receptor for FSH and they respond to the FSH, and the theca cell, they have the receptor for LH and they produce the androgen by the axon of LH, and the theca cell, they produce the androgen by the axon of LH, and the granulosa cell, they convert the androgen into estrogen. That aromatization occurs during folliculogenesis. So when the follicle is close to ovulation, this granulosa cells slowly they are developing receptor for LH. That is very interesting. The granulosa cell before during folliculogenesis, they are not showing receptor for LH, but when they are close to ovulation, this granulosa cell now they are expressing receptor for LH, and this expression of receptor is stimulated by the FSH and estrogen in the pre-ovulatory follicle. So when there is LH search and LH have the receptor on theca cell, and also the granulosa cell also develop the receptor for LH. So when there is LH search, this LH act upon the granulosa cell as well as theca cell, and they do so many activation of some of the mechanism for ovulation preparing. That is the starting stage of ovulatory process. LH search is very important for ovulation. I discussed during the folliculogenesis. So uh, LH search, it act upon theca cell as well as granulosa cell, because granulosa cell has also developed the receptor for LH in pre-ovulatory follicle or before ovulation. LH surge cause resumption of meiosis, or it helps the oocyte to complete the first meiosis division when there is LH surge. And this oocyte, the oocyte is frozen at metaphase, frozen at prophase 1, and that dictate state is maintained by the oocyte inhibiting factor secreted by the granulosa cell. And also granulosa cell, uh, they are preventing developing their receptor in them in the granulosa cell by secreting luteinizing inhibiting factor during the growth of the follicle. So as the follicle reach its develop, when the follicle develop or become large follicle, Graafian follicle, dominant follicle, then slowly the granulosa cell, as they are developing receptor for LH. So after LH surge, so this the component which is secreting by the granulosa cell, that is oocyte inhibiting factor, is prevented by the LH surge. And because of this prevention, the oocyte undergo meiosis 1 and complete its first meiosis division by releasing the first polar body. Then, as it is developing the receptor for LH, this granulosa cell, under the influence of LH, they are secreting the prostaglandins. And prostaglandins F2 alpha, prostaglandins E2, and prostaglandins A. So these prostaglandins play a very significant role during ovulation. So it prevents continuity of connective tissue of the theca layer, formation of connective tissue in the theca layer, and also vesicle formation is stimulated by the prostaglandins in the fibrocyte. And this is very important because fibrocyte, the vesicle that contain the hydrolytic enzyme, and that hydrolytic enzyme, it releases over the theca membrane, over the theca layer, and that this cause digestion of the collagenase matrix, which is over the theca layer, or over the surface of the follicle. And that is very important, formation of the vesicle and the release of digestive enzyme or hydrolytic enzyme that digest the matrix or collagenase, which is uh, over the follicle. And this diagram, the granulosa cell, which are very closely linked with the oocyte, which surrounds the oocyte very closely, that those cells are called, these granulosa cells are called cumulus cell. This cumulus cell have direct contact with the oocyte, and this cumulus cell secreting or forming the compound, that is cyclic AMP, CAMP. And during the growth phase, so when this cumulus cell or granulosa cell, which are very close to the oocyte, the cyclic AMP from the cumulus cell, it is transferred to the oocyte, and the oocyte, and then they are arrested at prophase 1, and this stage is maintained by the high amount of cyclic AMP. But when there is LH surge, this the production, the amount of cyclic AMP in the oocyte is decreased, and the oocyte complete its first meiosis division. So here, the cyclic AMP produced by the cumulus cell is that cyclic AMP, high amount of cyclic AMP, that maintain the arrested stage of the oocyte. When there is cyclic AMP drops within the oocyte, and immediately that oocyte completes its first meiosis division, and that decrease of cyclic AMP occurs at LH surge. So before LH surge and or before gonadotrophin surge, the oocyte is arrested at prophase 1 by the cyclic AMP formed by cumulus cell and transferred to the oocyte through gap junction. These are gap junction. This is this structure projections They have a projection direct to the oocyte, and through the gap junction they release the cyclic MP to the oocyte and maintain its arrested state. As the blood flow increases over the surface of the follicle, the leukocytes secreting the cytokines, and some of the cytokines like interleukin 1, they stimulate the cumulus cell which are very close to the oocyte, this cumulus cell. And under the influence of interleukin 1, this cumulus cell, they are producing very important factor, that is insulin like factor 3, and that is also take part in decreasing the cyclic AMP, means that cause cessation of the this supply of the cyclic AMP from cumulus cell to the oocyte. Therefore, there is a drop of cyclic AMP in the oocyte, and it completes the first meiosis division. So when there is LH search, so they cause drop of cyclic AMP so that the oocyte can complete its first meiosis division. And this cytokines produced by the leukocytes, and also they release the free radicals, and these free radicals and the cytokines, they also help in destruction of the tissue around the follicle, and this compound they cause inflammatory reaction over the follicle, and also they take part in the destruction of some of the tissue for during ovulatory processes. And activated basophil, monocyte, and neutrophils, endothelial cell, they are producing platylite activating factor. They are also taking part in inflammatory reaction and contributing the process of tissue destruction during fertilization. As the blood flow increases over the surface of the follicle, neutrophil macrophages also arrive to the area of the dominant follicle. And very importantly, the neutrophil, it bind with the receptor ICM, that is intracellular adhesion molecule 1, and with the vascular endothelium of the ovarian follicle, the blood vessel which are above the which are in the theca layer. So when neutrophil bind with the receptor in endothelium of the ovarian follicle, they releases very important compound, that is elastase. Elastase and other proteases. Elastase is a metalloproteinase. Metalloprotein are a structural protein over the surface of the follicle. This metalloproteinase digests those structural protein, this elastase, and also degrading the tissue around the apical surface of the follicle. LH search, we have so many proteases. Proteases means which digest the protein, and all the around layers around the follicle are the protein structure, extracellular fluid, extracellular matrix. So those matrix are digested by the many proteases secreted by the from the blood and other cells in the ovary. So that layer is digested by those proteases. And here we have the basement membrane. Basement membrane also contain laminin, collagenase type 4, and this is rich fibronectin and proteoglycans, which are major extracellular compound. And this compound also thought to this compound also digested by the gelatinase. These are also metalloprotein, proteinase, and plasmin or plasminogen activator factor. There's these proteases are also thought to digest the basement membrane. And basement membrane after or during ovulation process, this basement membrane is completely uh, degraded or destroyed, and the leftover after ovulation, the leftover structure become the corpus luteum, producing the progesterone. So, and over the and ovary also, the superficial layer of the ovary is the tunica albuginea. Tunica albuginea. Over the tunica albuginea, we have the epithelial layer. That epithelial layer also digested by some of the proteases. So, many proteases are released within the follicle, and from blood cells, and those cells they digest the superficial layer of the follicle. So here also very important the communication between the oocyte and cumulus cell. I told the granulosa cell which are very closely attached with the oocyte are called cumulus cell. So cumulus cell they supply the pyruvate for oocyte metabolism. So oocyte completely have to depend upon the cumulus cell. And oocyte also secreting some of the mitogenic factor which help for the proliferation and the development of the this cumulus cell. And very importantly, the oocyte is secreting bone morphogenetic protein 15 and growth differentiating factor. And this cumulus cell and the components secreted by the cumulus cell, secreted by the oocyte are preventing the cumulus cell from developing the LH receptor. I told you that granulosa cell developing the LH receptor, but the cells which are very close to the oocyte, those are called cumulus cell. The oocyte, they are releasing some of the mitogenic factor like this, some of these factors which prevent the development of the LH receptor over the cumulus cell. Therefore, this cumulus cell they don't produce the progesterone, but the cumulus that the granulosa cell which are close to the basement membrane, those cells are producing progesterone or they have the receptor for LH. They have so under the influence of oocyte, they are secreting some of the compound mitogenic factor. This cumulus cell they are secreting very important compound. This compound they form the cellular matrix or extracellular matrix, and that cause expansion of cumulus cell. This cumulus cell around the oocyte, those cell due to the development of the extracellular matrix, this cell expanded. It indicates that when the oocyte is matured or development achievement of the developmental competence, they secrete some of the component, and by the stimulatory action of the oocyte, this cumulus cell they are releasing or synthesizing some of the very important compound which form the extracellular matrix, and that matrix cause expansion of the cumulus cell, that indicating that the oocyte is maturing or oocyte has reached its maturity. The cumulus cell which are around the oocyte, this granulosa cell which are around the oocyte are cumulus cell. This cumulus cell under the influence of oocyte they are secreting the compound which form the cellular matrix. These are the cellular matrix between the cumulus cell and the compound, the hyaluronic acid, one of the compound secreted by the cumulus cell, and this hyaluronic acid structure are stabilized by the versican secreted by the mural cell. Mural cell is the granulosa cell which are very close to the basement membrane. The granulosa cell which are very close to the basement membrane are called mural cell. This mural cell secretes very important two compound. This is versican. This structure they bind with the one cumulus cell and with the hyaluronic acid, and they give the stability of the structure. This is CD441 receptor where hyaluronic binds one end, another end they binds with another cumulus cell with this receptor, and other compound and HC heavy chain. They are the protein. They are compound which are coming from the blood or serum or the plasma, and TSG96 also secreted by the cumulus cell and PTX3, the full form is given here. So this compound actually they make this extracellular matrix, and their stability, the stabilization of this structure formation and the stabilization or the structural this stabilization is necessary, and this compound play a very important role, and this compound also plays significant role during fertilization, because when oocyte is ovulated, the oocyte will escape from the follicle along with the cumulus cell, and during fertilization, when the sperm comes in contact with cumulus cell, they have to react with the extracellular matrix, and this extracellular matrix help the sperm to undergo capacitation. So that this compound also plays significant role in sperm capacitation. So here, this ADAMTS1, that metalloproteinase, this is a proteases. That proteases digest some selective portion of area of cumulus cell, and this yellow color, when this portion of the cumulus cell is the cellular matrix is digested, the oocyte is separated from that area. This is the cellular matrix between the cumulus cell, and this cumulus matrix is digested by this compound secreted by the mural cell, and because of this compound, they detach the oocyte along with some cumulus cell from a specific area. Suppose this area is digested, and this oocyte along with the cumulus cell are detached from it. As they detach from it, this oocyte move toward the apical area. This is the area. From lower part, it is moving to the apical part of the follicle. As we discussed, that the apical surface is almost digested by the many proteases. So when this fluid is giving pressure over this area, so you can find over the surface uh, raising the follicle, that area. Suppose this is the ovary, this is the ovary, and over the ovary you can find this type of structure, and this type of structure is called stigma. So the proteases, they cut this structure, and the oocyte is ejected out. Because of digestion of this area, the oocyte is ejected out from the follicle. So when this area is digested by the proteases, and the oocyte is released from the follicle, and that is called ovulation. So mainly many component and the changes of the receptors within the granulosa cell, and the component released by the theca cell or the blood vessels, the mostly the proteases, and the resumption of the meiosis and the developmental competence of the oocyte, and the stigma formation, and the proteases, they digest this apical surface of the oocyte. Therefore, the ejection of the oocyte occurs from the follicle. And that is ovulation. Thank you.